First of all, it's a real pleasure to come and share our story. Thanks to Rick and the organisers for inviting me. One of the sort of lessons from coming to these meetings and to other rare disease meetings is everybody in the audience pretty much has the same problems, the same ambitions, and the same set of learnings. And I think it's really great to hear everybody else's stories and sort of hear sort of echoes of what you've been dealing with for years. Okay, so as Rick said, I'm going to tell you about one particular disease, but we're a large organization spread over about 7 different universities across the world, including sort of Canada, Brazil, Germany, Sweden, UK, and the US. And we have the pleasure to work with about 9 pharma companies at the present, but we're an open science and non-patent, non-profit, charity-based academic research institution that's really targeted to understanding the basis of molecular disease and trying to find a way to catalyze early drug discovery. And, okay, so I wanted to try not to talk too much about science, but there will be a few science slides. But I want to try and echo the journey that we've been on, and I think probably everybody in this field sort of embarks on. And that's, first of all, what do we need to do to get from A to B? For us, we started with a genetic discovery for the cause of the disease. And so, you know, our first questions were, what's the disease mechanism? You know, what would be our therapeutic target? How would we go about that? And then once we have a target, how do we get to the clinic? And of course, that's something that Findacure are obviously there for helping a lot with in this space. So first of all, I'll tell you a little bit about the disease I've been working with, and this is purely by accident. I was based in the Institute, briefly, of Musculoskeletal Sciences in the University of Oxford, and that was purely an accident because our building was being put up next door, and we had temporary housing there. And the professors came to me and said, we've just discovered the gene for this disease, FOP, or to give its long name, Fibrodysplasia ossificans progressiva. And this is a disease that really is a germline mutation, so something you're born with, an inherited condition, but most of the cases are spontaneous, so a spontaneous mutation from the single cell embryo. And it can progress rapidly or quite slowly, but typically children 5 to 10 years old will start to see the first symptoms. And then by typically the 3rd decade of life, most patients are then in a wheelchair, immobilized, and then there can also be a reduced life expectancy. And the disease itself manifests in inflammatory swellings, which you can kind of see on the left in small children. And these are particularly related to trauma and injury. So bumps and bruises have an exaggerated effect. And these cause the breakdown of muscle and the infiltration of stem cells that then rebuild that space as bone instead of muscle. And this has a catastrophic effect on movement because once your muscle is replaced by bone, obviously you're losing the physical mechanics of moving, but also joints become locked. And so they could become statuesque in terms of being fixed in one particular orientation. And then there's a real difficulty in deciding, how do I want to be fixed? Will it be in a wheelchair or will it be in a kind of standing up orientation? Now, the most worrying aspect of the disease is that patients really struggle to get a diagnosis. They can often go 2 or 3 years without diagnosis, and they're typically treated as if they were cancer patients. People see these large lumps that arise on the body really quickly, and they obviously think these are tumors. We've got to get rid of them as quickly as possible, and that can have really bad consequences such as limb amputations, which are completely unnecessary because they don't have cancer, they just have a rare disease. Fortunately, the incidence is rare, so there's only 1 in 1 million. So in the UK, there are sort of 65-ish patients, and you can imagine that's quite a small, close-knit community. And so I'll tell you about how a small-knit community can get some research done. Okay. Okay, so this story really kicks off from patients themselves. So in Oxford was the National Centre for Clinical Care. So a lot of patients came to Oxford for their treatment, and typically they're only seen annually, say for an X-ray scan, to see what bone formation was going on in their bodies. But there was no treatment for them, and so patients would come and say, well, surely someone's researching this condition. You know, what would be done to help me? And you know, when the doctors say, well, no, I don't know anyone researching the disease, it's not the answer they want to hear. And so this lady, Sarah Cameron, said, okay, we've got to change that. Let's start a research group. And the clinicians in there said, okay, we'll help you set that up in Oxford. And so they started this University of Oxford FOP Research Fund, which has now sort of grown into a UK-wide patient group. And then sort of pretty much every country now has a patient group as well as an international FOP association. And so it was really the patients who said, we must have research, and how can we help fund it? And so this has been a trickle of funding, you know, with only 65 families in the UK affected. This is really a challenging area, but they've done really well in supporting, for example, in the first instance, a PhD student just to get research going. And then that's kind of triggered a sort of a snowball effect, and then that's gained attention of other benefactors and philanthropists. So quite soon along the way, this was really an international effort. Most patients with FOP are unlikely to have children, but quite remarkably, they found some families where there was a 3-generation family, and you could actually use that to identify the chromosome and then the gene linked to the disease. And this was published in 2006, which tells you this has been a long journey because obviously it's 14 years later now. And this disease was mapped to a gene called ACVR1, which I'll tell you on the next slide what it does. This is part of the TGF-beta family. So this is a gene, it's a receptor in the cell membrane, and it's really a receptor for ligands that are bone-inducing proteins, bone morphogenetic proteins. And so if you think of this as a bone disease, we're getting too much bone, and we have a mutation in a receptor that its job is partly regulating bone formation. So this kind of made sense. And interestingly, we had mutations just in one single amino acid in the protein. Okay. And so the question for us as a scientist was then, okay, what's the disease mechanism? What are we going to do to treat this? And I'm a structural biologist, so I look at proteins from an architectural point of view, and then look at mutations and say, okay, what effect will they have on the structure? Will they be disruptive and cause a loss of function in the protein, or will they do something interesting and activate them, and then we want an inhibitor? And at least from a structural perspective, this mutation looks like it disrupts things. Look, it should be a loss of function. It's basically breaking interactions in the protein. And when we look at similar mutations in other proteins, the genetic substitution itself is very commonly a loss of function event. And so with only one mutation, that was kind of my inkling, this is what probably happens. It turns out I was wrong, and this became clearer when further patients were sequenced. So the initial report was that only one mutation causes the disease. Once they identified what gene causes the disease, they could go out and then DNA sequence all the patients with the disease. And sort of internationally, this has now led to the discovery of maybe a dozen different mutations, all in the same protein, all in the same region of the protein, that give rise to the same phenotype. And it becomes a bit clearer when you see a whole pattern of mutations, a spectrum, than just a single mutation on its own. So we could understand a little bit more that all these mutations have a destabilizing effect. They're all regulating the on and off switch of this protein. But what our structure is of its inactive state, and they're breaking interactions in inactive state. And so rather than breaking things in terms of being destructive and causing loss of function, it's actually causing hyperactivation of the protein because we have an inactive protein, and it's just breaking it has a lock on it that would prevent it from signaling abnormally, and that lock is broken, so the protein is now doing things it shouldn't be, being overactive and causing this disease. So this is just one example where the yellow is the normal wild type lock, if you like, on the activity of the protein, but that gets broken when we get a substitution of this amino acid in pink at the bottom. And so we can test this in cells, so we can do functional assays, and we can see that if we look at this pathway, we can monitor how much this bone-producing pathway is active, and we can take that the normal protein under basal conditions where we don't stimulate it would have very little activity, but when we look at all the different disease-causing mutations, we can see an increase in activity, and that's been seen by groups all over the world, and we weren't necessarily the first people to do this. So that said to us, okay, we want an inhibitor. So that's our route to the clinic. And with others, we were able to develop tool compounds, put these into animal models. And so in a genetic mouse model, these will also get the human condition in terms of the disease appearance. And then if mice are treated with these tool drugs, we can block the onset of that disease. So this is really, really encouraging. So the next stage is, how can we really develop this to get to the clinic? And that, in some senses, has been a much longer journey. And I think going through clinical trials will probably be the next big challenge in terms of complete surprises along the way and lessons to be learned. So we were satisfied that we knew the disease mechanism and that we could go ahead to the clinic. It then turned out that there was a sort of trick that we hadn't understood. And this was published by a group at Regeneron, a big pharma company, particularly based in the US. And they discovered that the disease mutation actually did something extra in that while we have bone-making proteins, these BMPs, they're actually a family of about 30 different growth hormone-type proteins. And one of the set of these is called activin. And activin has a known role in regulating muscle mass. And it would normally signal through a completely parallel pathway, this TGF-beta pathway. So in normal people, it would only go through these red set of receptors. But in our disease mutant, suddenly it becomes promiscuous, and it starts signaling through this set of receptors as well. And you can see that in what some of you may or may not understand this, but basically the mutant has two sets of pathways that are active, and in a normal individual only have one. And what was interesting for that was that you could have an antibody to this activin protein, and that also cured the disease, at least in FOP mouse models. So this would be a scale where you look at how much bone is formed extraskeletally, and then this would be the vehicle arm or placebo arm, and then this would be the antibody-treated arm. Now, this solved a lot of interesting questions for us because the disease itself was always known to be linked to trauma and inflammation. If you have a bump or a bruise, you would get this trigger that would sort of accelerate the disease. And so activin is a known growth factor that's secreted in response to inflammation. So that kind of said something about what was going on with the disease. So I'll skip that. But this basically said, okay, there are multiple ways to target this disease. So Regeneron were developing an Anti-Activin A antibody, so blocking this growth factor, which in addition to these bone morphogenic proteins, is triggering the disease. We were interested in having an inhibitor binding directly to the receptor. So this would basically stop the stimulus, and this would stop it being signaled. And then other groups were looking downstream. So there are proteins that act on your DNA in terms of turning genes on and off. And so there were some vitamin A retinoid type ligands which can act on a DNA transcription factor down here that would block normal differentiation towards the bone pathway. And so we had 3 different strategies, and it turns out all of these are repurposing strategies. So as I'm going to tell you, we had an inhibitor for the receptor that was already been in the clinic. The Activin A antibody was already on the shelf in this pharma company, Regeneron, because they had been investigating it for muscle mass. And then this was a drug that Roche developed for emphysema, and then it was licensed out to a small startup who wanted to investigate it in FOP. So I'll come back to those other stories, but just to finish off my story, we were screening all the compounds that were in the clinic for FOP, for anything that would be effective for FOP. And we found this AstraZeneca compound, Saracatinib. And this is a really tight binding drug for our protein. And we could show how it works in cells and on the protein level. And then we could put that in mice. We could get inhibition of this bone formation in the FOP mouse model. And we could also see a slight dose response. And it's actually active at very low concentrations. I'll skip this, but it can also help with surgery to help patients remove. So if you're an adult patient and you have existing disease, allow surgery to remove bone formation. And so the final piece of the puzzle for us was just getting the funding. And this turned out to be very difficult. We probably wasted 4 years trying to do this. But we had some advantages in that the drug had already been in about 700 patients, both healthy and cancer patients, and also Alzheimer's patients. And so we knew everything about the dosing, and we had other pharma who were trying the other repurposing strategies who were then developing clinical protocols. And so we went to we went to everybody for funding, but we went ultimately to the EU. And this was really important because this is a disease with only 65 patients in the UK. There are about 3 trials already running. They're all taking all of the patients, so there's no patients. So you need to have a sort of international trial, and we've got a European trial with centers now based in Amsterdam, London, and Germany, just so that we can recruit patients from all over Europe. The first, with support of AstraZeneca, who are supplying the drug, we're hoping to get our first patients into enrollment within the next couple of weeks. And as an academic, you're kind of surprised at the timelines and what's involved in kicking off a trial. And we thought we were ready to go in December, and then we talked to the CRO who's actually making the tablets, and they want 3 months before they can actually deliver the tablets to the hospitals. And the reason is they want to agree the labeling that will go on the front of the bottle. And you think, okay, how do we wait 3 months for patients just to put a label on a bottle? But anyway, every step of the way seems to have something like that. So these are the specifics of our trial. We'll only really recruit 20 patients. I would really favor doing a trial without a placebo arm. There is a large natural history study with clinical recorded data that I think would potentially suffice. But all my other colleagues said, no, we really need to do the robust randomized control, placebo-controlled trial. And what we'll do is we'll treat for 6 months with a placebo, and then everybody will have the drug for a 12-month open label extension. And we'll be able to score the efficacy of this trial, both on the placebo-controlled 6 months and on the open label, just by comparison to natural history data. Of course, other things that need to go on will be translating that into every European language you can think of, and we're just putting up all the notices about launching the trials on the different disease foundation websites across Europe. We've called our trial STOPFOP for Saracatinib, Trial to Stop FOP. And we're quite fortunate in that the Scandinavian FOP charity is also called STOPFOP. So we didn't know that, but we kind of got some self-publicity there. And just to say, the endpoints will then be just volume of bone formed by CT. You know, this is very exciting for us. We have Regeneron as well with an antibody. But what's been surprising is once one company found out, oh, there's a route to the clinic, suddenly 10 other companies are all saying, okay, we're going to target this as well, we can see that there's a potential endpoint. It's tractable. When I entered this space, every clinician told me, such a pity we can never do anything for these patients. The first front runner, if you like, was this retinoid. And this was really just a sort of one-woman startup. And she sold it recently for $1 billion to Ipsen. And unfortunately, that's just been stopped, aborted in phase 3, partly due to lack of efficacy and partly due to toxicity. And then Regeneron's program is now about to enter phase 3. And then other companies have started to do primary research rather than repurposing research. And so those are a bit further behind. But a US company, Blueprint Medicines, has quite expertise in this. They've been having deals with Ipsen and formerly with Alexion to develop the kinase inhibitors, where I've got a repurposing kinase inhibitor, to take that into the clinic. I think they've just done their safety phase 1. And then I could probably tell you another 10 or so companies that are doing the same. And back to the comments this morning, the discussions were nice, and everyone hopes there'll be an approval. And if we get to that stage, then there are 2 lessons. One, how are we going to pay for it? But then also, all drugs are toxic. How can we give a drug daily to patients for 50, 60 years and not have side effects? And how can we treat children as young as 2, 3 years old? And that's an issue we still haven't got to the bottom of. And that will be what we learn from the clinical trials. And so maybe if we get lucky and we have 2 or 3 options, different drugs, each of which will have a different spectrum of side effects. If you could have holidays and switch from one drug to the other, that would be the dream scenario. Okay, and we'll skip that one. Thank all the people who've been involved. It's never one person. And thank also some of the funders who've really helped us push this through. Thank you.